Hello, my name is James Schnabel, and I'm going to be talking about how you can use the new connections between MaizeGDB and CoGe to learn more about your favorite gene. So for this example, I have already centered the Maize genome browser on a relatively random gene from the Maize filtered gene set, and let's say that there's some new mutation that I've mapped to this gene, or for whatever reason I'm very interested in it and I want to know more about its history. Was it conserved after the maize whole genome duplication? Is it conserved in sorghum? How is it conserved? These are questions that are really easy to answer within CoGe now that we've loaded in some of this data as links. So MaizeGDB now has this button called Click to See this region in CoGe, which does exactly that. It takes us to CoGe's genome browser, which is called Genome View. And because we came from MaizeGDB, we even get this little window that pops up and offers just a couple of links to the CoGe wiki, which will talk about the exact same things that I'm going to talk about in much more detail. You can see there's a very detailed walkthrough here, which is useful if you've never worked with CoGe before. But back to our genome browser. So you can see here is the same gene that we were just looking at in MaizeGDB, and because it has these large blue circles around it, you know that we were able to find at least an orthologous gene in sorghum. And if you click on these circles, or if you click on the gene model itself, it'll bring up the same information. But this GEVO link is a link to another tool within CoGe called GEVO that does a comparison of related sequences. And you can see in this case we identified a sorghum gene that was a syntenic ortholog, as well as two maize genes, one of which is the same maize gene that we're looking at right now, and the other is in the homeologous region of maize that was created during the whole genome duplication. So if we click very quickly on this, it will go to GEVO, which is automatically running a comparison of the two regions. It defaults to using BLASTZ, but you can configure the algorithm here. It's very customizable. And here you go. You can see we are able to find the gene both in sorghum and in this other complementary region of maize. So I believe this is our original copy we're looking at down here on the bottom. Here's the version in sorghum and the other version in maize, and you can see maybe the one we're looking at has some exons that aren't conserved in sorghum in the other version of maize. So this could represent an annotation error. It could represent something really cool biologically. If you click on the individual hits, you can see details. This maize region is very conserved at 96% identity, which is very relatively high for these things. What you'll also notice is that, for example, this gene right downstream from the gene we're looking at is only conserved in this one maize region and not in the other. One other thing I wanted to show is that you can click this button called Tandem Duplicates, and this is also in the process of becoming publicly available. And you'll see this gene over here is marked in blue, and that shows that this gene was found to be in a tandem array of three nearby genes. So if we scroll along a little further here, you can see here's one of the other tandem duplicates, and here's a third one here. And these were actually found to be syntetically conserved in sorghum, and you can see this one sorghum gene was found as a match to both of those. So what we're looking at here is what the new links that we'll be deploying in the next couple of days will be able to do, where they actually predict the region that a gene is expected to be found in. So in this case we're looking at the yellow gene, which is conserved in one maize region as well as in sorghum and not the other. And if you look at the blast hits from this region, they go up to this gene, and you can click here's the last one on that end. And then there's this big gap of sequences that don't hit, and then there's only part of this gene. And you can see that these genes match up almost exactly against each other. So this whole piece of sequence is completely missing from this maize chromosome, and this is actually only 300 some base pairs, whereas the whole gene, when we look comparing the sorghum to the other maize homolog, is 680. So this is a gene fragment that's been conserved in maize, which I think is relatively fascinating that we can see these leftover chunks of dead genes. Well, this is the end of this brief tutorial. I hope that this has at least whet your appetite for some of the comparative genomics things that are now made possible within CoGe and our collaboration with MaizeGDB. And have a good night or morning or whatever time it is when you're watching this video.